Welcome viewers. Today in this session we will talk about the different structures of DNA which are found inside the cells of living organisms. The structure of DNA can be divided into four main parts, that is the primary structure, the secondary structure, the tertiary structure and the quaternary structure. Now, before we move into the structures of DNA, we should first know what are nucleosides, nucleotides and tautomerism of bases. Now, first let us see that what are your nucleosides. The nucleosides are generally nothing but it is an addition of a base to a sugar. Now, the bases which are heterocyclic in nature are added to the sugar with the help of a covalent bond. The covalent bond in this case is known as N-glycosidic bond. Why? Because the bond is formed between the carbon atoms and the nitrogen atom of purine or pyrimidine. Accordingly, it is known as N-glycosidic bond. Now, since there are four different types of bases found in DNA, there can be four different types of nucleosides. They are deoxyadenosine, deoxycytidine, deoxythymidine and deoxyguanosine. Now, accordingly, the nucleotides are phosphorylated derivatives of nucleosides. When a phosphate group is added to the C5' carbon atom of a nucleoside, then it becomes a nucleotide. So, we can say that a nucleotide is 5' phosphorylated derivative of nucleosides. Again, there are four different types of bases found in DNA. Accordingly, there can be four different types of nucleotides. So, DNA is nothing but a polymer of polynucleotides. So, it is this polynucleotides which are joined in a polar manner or end-to-end manner with the help of strong bonds which results in the formation of polynucleotide chain. Now, before we move to the primary structure of DNA, we should first know that what is tautomerism of bases. Tautomerism of bases is an important property of the nucleobases. Now, since the nucleobases are aromatic rings and they consist of alternating or double bonds and hydrogen atom, so there is a shift of the hydrogen and electron clouds within the aromatic rings, resulting in the formation of alternating structures. The structures are known as tautomers. For example, the amino group found in adenine and cytosine are capable of forming the imino form. Now, since imino form is highly unstable, so it is formed during a very short period of time. Accordingly, the guanine and thymine, which contains a keto group, is capable of showing tautomerism and gets converted from the keto to enol form, resulting in the change of base pairing potential. With the tautomerism, it leads to the spontaneous mutation found in the living organisms. Another characteristic property of the nucleic acids is that it is able to absorb UV light. This property is due to the presence of nucleobases which have conjugated double bond system within the ring. As a result of the conjugation, they are able to absorb the UV light maximum in the region of 260 nanometer. So, hence, because of this property, we are able to quantify nucleic acids. Now, let us move to the structures of DNA. The structures of DNA can be in the form of primary structure, secondary structure, tertiary structure and quaternary structure. Now, let us first know about the primary structure. The primary structure is nothing but a polymer of nucleotides. So, it gives information regarding the sequences of the bases, like the four different types of bases, in what sequence are they present in the given DNA molecule. Now, each nucleotide is joined to the next nucleotide by the means of phosphodiester bond. The phosphodiester bond are strong covalent bonds. and it is because of this phosphodiester bond that polarity is imparted to the polynucleotide chain. Now, how are the phosphodiester bonds formed? Now, first, suppose if this is a nucleotide. So, it is joined to the next nucleotide by the means of the phosphodiester bond. The phosphodiester bond in this case is formed between the 3' hydroxyl end of the 2-deoxy D-ribose sugar with that of the 5' phosphate group of the incoming nucleotide, and it imparts a polarity. What is the polarity? So, you can see that at the end, now since it is present at the 5' end of the carbon, so we can say that this is a 5' phosphate group, and since it is containing a hydroxyl group at the 3' end, we can say it is the 3' hydroxyl end. So, this is nothing but it gives directionality to the polynucleotide chain. So, again in this diagram, as we can see, the polynucleotide chain which is consisting of more than 2 bases. Again, the same polarity is here, the 5' end and the 3' end. And the primary structure mainly gives information regarding the sequence of bases. The secondary structure of DNA, or the DNA double helix, which was first elucidated by Francis Crick and James Watson, represents the secondary structure of DNA. Now, what are the features of secondary structure of DNA? The first feature is the secondary structure of DNA is formed, or it is made between 2 polynucleotide chains. Now, these 2 polynucleotide chains are wound around each other in a right-handed manner. The 2 polynucleotide chain are anti-parallel in position or in directionality. That is, the 2 chains face opposite to each other. The bases in the 2 chains are complementary to each other. Now, the question comes in, why the 2 chains are anti-parallel in nature? It is a direct consequence of stereochemical result. Now, since the bases generally face inwards and they form hydrogen pairs, so when the 2 polynucleotide chains are in antiparallel fashion, maximum number of hydrogen bonds are found or formed between the bases. The hydrogen bond gives stability to the DNA double helix. As a result, the 2 chains are antiparallel in nature. The third or the last category or the property of DNA double helix is the sugar phosphate backbones generally face outside, or they are present outwards of the helix. Now, why is it so? Because the sugar phosphate backbone, being hydrophilic in nature, interacts with the aqueous environment of the cell, while the hydrophobic bases, they point towards the interior of the double helix. As you can see in this diagram, this blue ribbon which is shown by the ribbon figure, so here this represents the phosphodiester backbone, while the bases are facing inwards. Now, next, the bases or the set of base pair is highly fixed. So, we know that there can be only 2 types of base pair, that is add A and T and G and C. Now, why there cannot be any other base pairs like G and T? It is because the A and T are hydrogen bonded to each other, or the functional groups present in adenine and thymine participates in hydrogen bond formation. So, as shown in this diagram, we can see the thymine which contains a keto group. The keto group present at position 4. Accordingly, it forms hydrogen bond with the hydrogen of amino group of adenine. So, in this case, we can see that adenine and thymine are stabilized, or the pair is stabilized by formation of 2 hydrogen bonds. In case of G and C, it is stabilized by formation of 3 hydrogen bonds between the functional groups. So, that's why it is said that the GC pair is more stable than AT pair, as it has got 3 hydrogen bonds. Now, if any other set was there like G and T, the hydrogen bond formation would not have taken place, As a result, in nature, we only find A and T and G and C and no other base pairs. Now let us move to the other property, like why the diameter of the DNA helix is 20 angstrom, or why it is 20 angstrom uniformly. Why not? It can be 18, it can be 19. Why only 20 angstrom? Now it is due to the reason that the distance between the C-1' carbon atoms of AT pair is approximately or around 1.1 nanometer. Again, the distance between the C-1' carbon atoms of G and C pair is 1.1 nanometer. So together with the sugar phosphate backbone, it adds up to 2 nanometer. Now, had the base pairing taken place between two purines or two pyrimidines, the diameter of the DNA double helix would have varied. But since the base pairing always takes place between a purine and a pyrimidine, it gives rise to a uniform diameter of 20 angstrom or 2 nanometer only. Now, the next feature of the secondary structure is the base pairs are separated from each other by a distance of 0.34 nanometer, which is also known as the rise of the helix. Now, a complete 360 turn of the helix, it measures about 3.4 nanometer, which is also known as the pitch of the helix, which represents the length of one complete turn of DNA. Now, in one complete turn of DNA helix, there are 10.4 base pairs, and each base pair is rotated from the next one or from the adjoining base pair by an angle of 36 degrees. So, the base pairs are not fixed in position. One is rotated in relative to the next base pair. Why? Because the backbone of the DNA helix or the sugar phosphodiester backbone is what it is helical in nature. So, in order to accommodate the base pairs which are of fixed length, it has to be rotated. Now, if we look at the DNA molecule, there are grooves found. One is the major groove and the minor groove, occurring along the length of the double helix. Now, what are major groove? Major groove are formed when the distance between the two phosphodiester backbone are large, whereas the minor groove are formed when the distance between the two phosphodiester backbone or between the backbone are less. Now, it is formed because of the geometrical consequence. The geometrical angle which the glycosidic bond of the purine or pyrimidine makes with the sugar phosphate backbone. So, it is generally thought, or it is now known, that the geometrical angle of the purine is 240 degree while the pyrimidine is at 120 degree, which give rises to the formation of grooves. Now, it is seen that generally the DNA binding proteins, it binds to the major grooves. Why? Because the DNA or the major groove is rich in chemical information than the minor grooves. Now, since it is rich in chemical information, so most of the bases can be known or read out from the major groove, and hence the proteins can form or interact with the specific functional groups present on the bases in the major grooves. As a result, most of the DNA binding proteins, like the proteins involved in transcription, replication, repair, recombination, bind mostly to the major groove. While the minor groove, it allows the addition or it allows the incorporation of small molecules like the dye molecules or sometimes the histones, which are known to bind to the minor groove. We must know what are the factors which are stabilizing the DNA double helix. Now, the first energy or the force which is stabilizing the DNA double helix is hydrophobic interactions. The hydrophobic interactions are formed between the stacked bases. Now, since the bases are aromatic rings, so when one base is stacked over the other, so hydrophobicity increases. So, these are known as hydrophobic force, which allows the base to be stacked on one top of each other. The interactions or the force which contribute to the stability of the DNA double helix is stacking interactions. Stacking interactions are found or formed when two atoms are brought in close proximity to each other, also known as van der Waals forces. The third point is when hydrogen bonding takes place between the base pairs. So this contributes to the formation of energy. Then we have ionic interactions. Ionic interactions taking place between the negatively charged sugar phosphate backbone with that of the positively charged ions such as magnesium ions. And the fifth part is the covalent bond. The covalent bond found between the atoms of the bases. Apart from that, we have the N-glycosidic bond, the phosphodiester bond formed between the two nucleotides. So all of these, they release energy, thereby in total lowering the free energy of the DNA double helix and thereby stabilizing it. Now another characteristic property of hydrogen bond is that since the hydrogen bond are found only between adenine or thymine or guanine and cytosine, so it imparts specificity to the base pair. Because of the property of the specificity, so the DNA is a self-encoding in nature, or we say that the two DNA chains are complementary to each other. As a result, the DNA replication or the mode of DNA replication is semi-conservative in nature. So if you know the sequence of one strand or if you know the sequence of bases present in one strand, you can know or decipher the sequence of bases present in the other strand too. Why? Because the two bases have to be complementary to each other since they are pairing by hydrogen bonds. In this diagram, as you can see, the bonds have been shown which are imparting stability to the DNA double helix. One is the covalent bond, the hydrogen bonds and the hydrophobic bonds. So we can say that the DNA, it consists of an hydrophilic portion present on the outside and there is a hydrophobic core as we can see on the inside of the DNA double helix. Because of the presence of hydrophobicity of the stacked base pairs, so water cannot enter inside. So it increases the enthalpy or the entropy rather. So when there is an increase in the entropy, the formation of that molecule is favored. Now let us know that why the backbone of the DNA is not a straight line. It can be had or the DNA molecule could have taken a structure like this. So in this case you have the base pairs which are stacked inside and the straight hydrophilic sugar phosphate backbone. But as we can see that the DNA is nothing but it is helical in nature. So why the backbone becomes helical? It is to promote maximum number of base stacking and the maximum amount of hydrogen bonding which can only be made possible if the sugar phosphate backbone is made helical. So when it becomes helical, the two base pairs are brought in close proximity which promotes hydrogen bonding and the base pairs are more stacked over each other, promoting stacking interactions. So that is the reason why the backbone of the DNA double helix is helical and not as a straight line. X-ray diffraction studies were made on DNA fibers. It was found that 11 distinct conformations of DNA can be found inside a living cell. The type of conformation or structure which a DNA takes up depends upon the base composition of DNA as well as on the physical environment. As you can see through this diagram, the aDNA is what it is. We can say the aDNA is formed when the bDNA is compressed from the top. So resulting in a formation of short and squat form. So the A form is short and squat form of bDNA. Whereas the zDNA we can say can be formed when the bDNA is pulled longitudinally. So the zDNA represents the longer and thinner version of bDNA. Now, if we study the cross-sectional view of A, B and Z DNA, we find that the most ordered secondary structure is that of B DNA. And the A DNA, it has increased diameter and there is a presence of a hole in the center of A DNA. While the Z DNA is more zigzag in orientation and they are not present in proper way.
Ma'am, in case of A DNA, how does the space that we see in the cross-sectional view affect the stability of the molecule, and under what conditions does this DNA generally form?
Very good question. So, what is happening is the A DNA or generally the B DNA is more stabilized form. Why are we saying it is the most stabilized form? Because a number of forces are acting within the B DNA molecule to stabilize it. Now, when the DNA is placed under dehydration condition, then A DNA or formation of A DNA is promoted. So, A DNA is generally formed in bacterial endospores or in fungal spores. Now, since in A DNA, what is happening? The water, there is less water. Now, there are two kinds of functional groups or compounds which have got high affinity for water in DNA. One are the phosphate backbone, another are the bases. Now, since bases are more hydrophobic than phosphate, it has got less affinity for water. So, it is the phosphate which forms an ionic interaction with water. Now, when the water contained decreases, then the A DNA is formed because the phosphate then present on the outside forms narrower and deeper grooves. The major groove becomes narrow and deeper, allowing more water molecules to interact with the phosphate group. As a result, the hydrogen bonding between the bases we can see are disrupted as the bases are displaced from each other, which results in the formation of a hole when we see it from the cross-sectional point or from the top view. So, this is not so much stable as compared to B DNA. And it is formed only under certain circumstances or under particular physical conditions of the cell. Now, let us come to the different structural properties of A DNA, B and Z DNA. So, what are the first property? First, as we know that A and B both are right-handed in nature, whereas Z, it is left-handed in nature. The A and B DNA represents more structured molecule than Z, as the name Z is derived because of the zigzag nature of the sugar phosphate backbone. The diameter of the DNA helix is 25.5 angstrom, 23.7 angstrom and 18.4 angstrom in case of A, B and Z respectively. The rise per base pair or the distance between the two base pairs are 2.3, 3.32, 3.8 angstrom in case of A, B and Z DNA respectively. So, another important features is the number of base pairs. In case of A DNA is 11, in B DNA it is 10 and in case of Z DNA it is 12. So, in this way we can differentiate the different structural features of A, B and Z. Another important property is the length of the pitch. The length of the one complete turn of the helix is 24.6 angstrom in case of A DNA, 33.2 angstrom in case of B DNA and 45.6 angstrom in case of Z DNA. So, that is why we are saying that when B DNA is compressed, it gives rises to A DNA. We can think in that way, and when B DNA is pulled longitudinally, then it becomes linear and thinner as seen in Z DNA. Now, let us move to the other non-conventional structures of B DNA. So, what we have thought is B DNA is a long molecule, moving or rotating in the right-handed way. But it has been found that there are certain other DNA structures or secondary structures which are formed depending upon the base composition. Like when they are palindromic sequences, it gives rise to cruciform structure rather than the B DNA form. When there are polypurine or polypyrimidine nucleotides, then triplex DNA is formed. Again, when there are mirror repeats or direct repeats, then other structures of DNA are formed. Now these structures of DNA are generally, they play a role in recombination and other repair processes. So these structures of DNA are generally recognized by enzymes which initiate or facilitate this kind of downstream processing. So it is not always that the DNA has to be in a B form from right to the beginning to the end. So as you can see, this is a triplex DNA and it deviates from the normal B DNA which we find in the cell. The triplex DNA, why it is named so? Because it is made up of 3 strands of DNA. It is favored only when there is a continuous sequence of homopurine, homopyrimidine, or there are phased A tracts likewise. So the structure of DNA or the secondary structure of DNA entirely depends upon the base composition and on the physical environments of the cell. Now let us move to the tertiary structure of DNA. The tertiary structure of DNA involves a higher order folding of regular secondary structure. So in this case, the tertiary structure represents the supercoiled DNA, which is found when the DNA becomes covalently closed circular in nature. The two terms are linking number generally associated with tertiary structure. Now let us move to the quaternary structure. So what is the quaternary structure? It involves the higher order folding of chromatin in chromosomes. So when two different DNA units are interacting with each other, it forms the quaternary structure. DNA is one of the most stable forms in the cells of living organisms. The structure of DNA involves the primary structure, which gives information about the sequence of bases, secondary structures which help us to know about the function of DNA or what are the functions of DNA in a cell. The third structure or the tertiary structure is formed when DNA has to be compacted in a very small volume. So the supercoiling takes place, and the quaternary structure which enables it to function as together as an integrative unit. The structure of DNA which it will take up depends upon the base composition and on the physical conditions of bases, and depending upon the structure of DNA, it participates in the different downstream processes as replication or transcription or recombination or in repair processes.